If you didn't think of those things as you parse through the intro and the discussion, that's okay. This is, again, a skill that comes with time and practice. We're going to leave to now explore the second component of the paper anatomy, which is the overall experimental design and the materials and methods. We're going to look for limitations in them. Now, every scientist makes what I have in parentheses here, choices. There are a lot of variables in any experiment, especially in one like this where we have humans involved and all sorts of environmental factors to consider. So when you're setting up your experiment, as I mentioned in the first video, it's very important to define your parameters, establish your controls, so that you know when and where you can trust your data and make an important inference from your data to know what those results are suggesting towards what you're studying overall. The scientists performing the work at the bench after the design is completed make choices as well in terms of how much they're pipetting, or if they don't see a result, how to troubleshoot it. Maybe there was some user error, maybe there was some mechanical error. How do you figure that out and how do you move forward? We make choices. Those choices can limit us, and that's both good and bad. We have to live within the limitations of our choices. We have to live within the limitations of our models, even the mouse models that I just talked about a moment ago. So the most we can do is establish a very, very well-controlled study and perform well-controlled experiments. That's our job as a scientist, and we're going to learn how to do that later on in the semester. So let's move to the materials and methods, and I'm going to start right there with the very, very first portion of it. And the materials and methods on the second column of that last page there start with population enrollment. Something I love, fecal sample collection, and DNA extraction. So these were the very, very first steps in sort of conceptualizing this work, establishing how the poop was going to be collected, and of course the poop is where you find the gut microbes, and how the DNA was going to be extracted from those poop samples, the DNA of the microbes, so that they can be identified for the bacteria species that we learned about in this paper. Reading right off the top, it says, we enrolled 15 healthy children living in the rural village of Bull Pond district of Nanoro. And I'm going to pause right there, not because I'm struggling to read those words, but because of the number 15. You may have noticed that on the very, very first page of this paper, under the section marked Results and Discussion, the very first section is called Characterization of Dietary Habits of Children from this area. I'll read that first sentence to you. It says, in this study, we characterized the fecal microbiota of 14 healthy children from the Mazi ethnic group, BF for short. And then it goes on to say, and 15 from healthy European donors. So that right there is a discrepancy. This says in the methods 15 healthy children from this group, when actually it was 14. We know it was 14 based on some other evidence, and I'm going to go through that with you right here. And so hopefully this is just a typo, but your spidey senses might have been alerted if you looked at the methods and saw, that doesn't match. This is not correct. So hopefully this was just a typo. It says that you can look at table S1 for some more information. And I'm wondering if you actually clicked on the supplemental materials. Table S1, where the S stands for supplement, can be found in those supplemental materials. And so you've had the PDF of this through the Canvas platform. And if you scroll down there, there's a link. And if you click on that link, you can see all the supplemental materials. Those include an expanded methods and some tables that further explain the patient donors from both groups, as well as the diet and other environmental conditions. So let me show you that. Here is table S1, which I've copied from the supplement and put right here for you. And on the left column, you see ID, and so they've given an anonymous number to each donor from both groups, BF and EU, designating the different areas, Burkina Faso and Tuscany, Italy. So if you were worried about that discrepancy and you wanted to make sure that you had the right number, this is one place to find it. And so, as you can see, there are 14 BF samples listed in the chart and 15 from the European group. Therefore, we do think this, plus 29 being the number of total donors indicated further on in the text and in the figure legends, etc., we're okay assuming that it was just a typo there in the methods, and that everything was 14 BF and 15 EU samples in this particular study. What's more interesting to think about is the last two columns. In one, the route of delivery, and two, the number of months without antibiotics. And it's fantastic that you have this data here, but it certainly does pose a limitation, and they acknowledge it as such when they're saying that we can't definitively go to causation, that there's other variables at play here potentially. And these are definitely two of them. So we know now, through the wonderful world of microbiology in primary literature, that the route of delivery affects the microbial composition that you get early in life, whether you're born naturally or via cesarean. And you do see a couple of instances here where children were born via cesarean. We know that breast milk and breastfeeding and the amount of breastfeeding one gets do change that. And so there were some differences between donors in that as well. And then, of course, antibiotics. And so this is a significant one in that the BF samples never were exposed to it, and the EU were, or at least for some period of time. And so these are very important limitations to make you think that there's potentially more than just diet at play when it comes to characterizing the microbiota from these groups. So there's a lot to dig in here into, and we might get into some more of these things in class. Doing great. Let's move on to the next section of the materials and methods. And again, we're not going to get into every single one, but I'm going to give you a few because we're again thinking about limitations based on choices. So this is 16S sequencing for rRNA gene amplicons. And so this is a very, very important choice. And in the world of sequencing, we are trying to determine identity. And there's two ways we could do that predominantly for microbes. One is to look at this 16S region. 16S is an indicator for something that all microbes have. And this particular region contains both conserved regions that are similar for every microbe, and then variable regions that make each microbe unique, and we could use to sequence and tell who's who. They're kind of like the barcoding regions of the 16S. And so that's what they mean by V in this sentence. V stands for variable, and that's what 16S is, where we're focusing on the 16S RNA region. And so I'm going to put those in boxes right here. The V5 and V6 hypervariable region. It just so happens that there are 9 variable regions within the 16S region, and they're only going to look at 2 of them, V5 and V6. Now, there's reason for that, and that's because a lot of the gut microbes fall within those 2 regions, 5 and 6, for their barcoding abilities. However, we're also excluding then bacteria that would be sequenced if we had probed, say, variable regions 1 and 2. We're not going to come up with those, and maybe those are actually key species that are determining differences between these 2 diet groups, but we just wouldn't recover them. Same if the key differences were actually in variable regions 7 or 8. We're not going to find those because the authors chose to focus on simply these 2 regions for their sequencing, V5 and V6. And so that's a limitation. It's based on good data, but it is nonetheless. Now, this paper was published in 2010, and fast forward now to today, and there are another way you could actually do the sequencing analysis for microbes, and particularly for bacteria. and that is to do a whole genome approach by something called shotgun sequencing. The main difference is between the 16S and the shotgun is that in the 16S approach, we're again targeting barcoded regions within one part of the bacteria chromosome. And in this case, it was even further down the rabbit hole, targeting V5 and V6, just those two regions. Shotgun sequencing chops up the whole genome and looks at all the DNA, and therefore would include all of those variable regions and other regions of the chromosome too, with the added bonus of potentially giving us some predictors of gene function. So this is an advanced technology. Both of these have strengths and weaknesses associated with each, but you know what, that's for another class. But I just wanted to give you guys a heads up as to how the study was done, and that this represented an important choice that then leads to some limitations in what you see. Now here's where that plays out a bit for you, and I'm going to actually, even though I'm still focusing on the methods, I want to look forward a little bit into the results, and really it's hard to separate the two. So the choice of methods and the limitations in design certainly affect the results, and here's that example then playing out for you, where the V5 and V6 sequencing decision affects the depth of what you see when you're looking at the composition of microbiota between the two groups. And so right here is figure 1, and I actually copied this over from the paper and put 1A side by side with 1B. And I'm going to indicate to you by showing this doubled arrow line that our resolution of sequencing only gets us to the phylum and genus levels here for the bacteria of interest. And so we don't actually get to species. I don't see anything beyond Prevotella, for example, as the genre, or Bacteroides or Rosebaria. These are some of the example genre. The phylum level is the Bacteroides and the Firmicutes. So we can't see anything more depthful. For example, on the right in 1B, in the EU group, it says Bacteroides, the second one down after Allostipes. The Bacteroides genus contains a lot of species that may or may not inhabit your gut, and one of those is Bacteroides fragilis, which I always abbreviate in my teachings as BF, your best friend. It's just one of those bacteria that we know to now mechanistically train your immune system, keep inflammation low, and keep you healthy. You want to have that BF. We also know that not all BF are the same, and so there are also differences between genus, species, and then strain. So Bacteroides fragilis strain 1 or Bacteroides fragilis strain 2, and the difference between strains would be at the genetic level, and there's very important differences that potentially play out in composition of gut microbiota in humans. And so if all we get is to the depth of the genre here, and we don't even have the species, then we definitely don't know anything about the particular strains these people might have. And it's important because not all Bacteroides fragilis are the same. Come out to your skin. Not all Staph aureus are the same. There are different strains that live on your skin. In fact, in any given community of organisms in any given location, whether that's skin, gut, or soil, we can't assume that every single cell of a particular genus and species are exactly the same strain. So it's true that the shotgun approach would have gotten us a little bit closer, a little bit more depthful towards strains, but again, there's limitations, and so we're not just going to make that blanket statement just yet. Let's go down further then in the methods to the next section, quantifying and comparing diversity between BF and EU populations. And I really want to point this one out because it will be very consistent with things you've heard before in this class. Right off the bat, how do they analyze their data? Well, differences between populations have been analyzed using parametric ANOVA and something called non-parametric Kruskal-Wallis tests. Okay, ANOVA is something we focused on and I hope that's gotten your juices refreshed. There are a couple of quiz questions this week asking you to draw back on your material and your knowledge from that particular week. For example, you're starting to think about normal and non-normal distribution and whether or not this matters at all, right? Yeah, sure you are. Yeah. I can see you nodding. I know. That's good. So that's one component of it. Later on, they indicate that the non-parametric rank-driven Kruskal-Wallis test was preferred for some things. And it says which performs well in absence of distributional assumptions. So we're going to explain some of these things in class, and there's actually a piece of your homework that asks you to look into this further, not just that quiz question. But these are important decisions. These are important choices based on the data they have and why they actually chose to use these. And so what we're hoping now is that you're really starting to feel comfortable if we kind of think back to what this class has been to this point, as we're almost near the halfway point. So we've done data visualization, we've done organization of it, we then analyze that data via different statistical methods, and now we went to ANOVA. This reading of the paper and analyzing of the paper is also pulling in those lessons. As you can see, scientists making choices and publishing their work based on how their data is indicating they need to do that. Okay, so you're doing a great job, and we've crossed off 2 of the sections. Let's move to the 3rd, and that would of course be the results. Again, we haven't hit them all, but that's going to be part of our class exercise this week. I'm going to actually go ahead and fast forward to figure 3. And I've got here the figure legend for the entire figure. Short chain fatty acid producing bacteria could help to prevent establishment of some potentially pathogenic intestinal bacteria. And I have part A illustrated for you here. And so what you see is on the y-axis, short chain fatty acids in fecal samples given to you as a particular concentration, micromolar per gram. You have a black set of bars and a gray set of bars where those colors differentiate the diet groups, black being the BF and gray being the EU. And then you can see comparisons for, on the x-axis, total short chain fatty acids, or each of them the 4 individual ones that follow: acetic, propionic, butyric, and valeric. As you can tell by the asterisks, I actually don't know. I always biff on that pluralization of asterisks. Say it how you like. There's either 3 or 2, but in any case, you can see at the bottom that the p-values based on one-tailed student t-tests all fall in significant ranges, where in this case you've got 2 or 3 in figure 3a are all either less than or equal to 0.01 or 0.001, depending on if it's 2 or 3 respectively. That's interesting. One of the things I've taught you in the first video to think about is looking for sentences or statements in the results that don't quite match the actual figures. This happens to be one of those times. And so here is the text from the page that follows the figure where the graph is actually being referenced. And so I'm going to box this part right here. It says, whereas acetic and valeric acids were comparable in both groups. Let's read that whole sentence. In particular, propionic acid and butyric acids are nearly 4 times more abundant in BF than in EU fecal samples, whereas acetic and valeric acids were comparable in both groups. That word comparable suggests that they're the same and that they wouldn't be significantly different. However, if you look at the graph for both acetic and valeric, you do see that the diet groups are differentiated by asterisks that indicate statistical significance. So this actually doesn't quite line up, and this would be a experimental critique related to the results as written. As a quick aside, related to bar graphs and showing data in that representation, and we've talked about data visualization, I'm giving you a reference here at the bottom. That's the DOI number for a paper. And this is a really, really nice paper that actually looked at how particular probiotics protected against Vibrio cholera infection in the gut. And the reason I like this presentation better than bar graphs, which they could have absolutely given you, is that you see all the individual data points and their distribution within each group. So on the x-axis, you see V, which is how much Vibrio you'd get. And the second group, V plus L, is how much Vibrio you'd get if Vibrio and a probiotic marked L were added together. So there's obviously a significant difference between the blue and red groupings. The line indicates the average for each of these sets. And so not only can you count the number of data points in each, so 1, 2, 3, 4, 5, 6, 7 for blue, and 1, 2, 3, 4, 5, 6, 7 for red, that's consistent. Our N is the same. And of course, looking at sample size and consistency between sample sizes is certainly something we're thinking about, as mentioned in video 1. This is really nice because it gives you the entire distribution, and you can see, for example, where some data points overlap with other data sets. So look at those two red ones overlapping with some of the, including the average values in the Vibrio group on the left, or the blue ones. And that's nice to see sort of that biological variation and have an appreciation for that. It's not simply all or one, all or nothing. You have some data points that do indeed overlap between groups for especially things like, in this case, a mouse study where each of those circles is a different mouse. There's a lot of biological variation to consider. So it's nice to see it represented. So here's an example where it's hard to tell whether or not the sentence as written and the results as given are in alignment with each other. Where we're going to look is right above figure 3. We're going to go to the bottom of that left column, just above it. And I'm going to blow that up for you here as indicated by the arrow. We're going to focus in on the section that says microbial richness and biodiversity. So we then compared the richness as estimated by the Cow1 index and the biodiversity assessed by a non-parametric Shannon index for the two BF and EU groups. In our calculations, we took into account different OTU distances. Fantastic. And then it says, using the non-parametric Kruskal-Wallis test for comparisons, we found significant differences, I'm going to blow that up for you, in both richness and biodiversity between these two groups. The question is that actually what the data does show you? When you look at this S6, it's a bit hard to really see what's happening. I'm not going to put the box plots from figure S3 that do show the Shannon indices generated from non-parametric analysis. The thing that's confusing is that when you look at this table, which doesn't reference in the text NP Shannon indexes, they just refer to those in figure S3. You see in the last 3 columns NP Shannons with p-values that are not significant. So it's very hard to reconcile then sort of this discrepancy between those two things. And sometimes you see that, and there's valid reasons for this, but we can't really make that determination given the two different presentations of those data sets.